Okay, I don't know if there's any coffee or tea left. Feel free to help yourselves if there is. And is it not going? It's not going. Okay. While we're trying to figure out some of the technical issues, let me just go back and give you sort of a different frame of reference related to Zika. So right now, when we're taking a look at the Zika virus and what's going on in Latin America, and it's moving northwards, um, if you read the Lancet Journal article that came out yesterday, one of the reasons why we're so concerned about this, and I'll show you a statistic related to this later. If you have an infection right now and you're pregnant in your first trimester, usually the rate at which microcephaly occurs during these types of infections is relatively low. It's like 1 in every 3,500 to maybe 1 in 10,000. According to the Lancet article that came out yesterday, if you are currently infected and have Zika virus, the odds that you will have a baby that has microcephaly is 1 in 100. And so you can see that there is a clear and present worry about this whole situation that's going on in Latin America. And this gets into some of the issues that we're going to be talking about today related to some of the different things that may result in microcephaly. We're not sure. This is brand new. It's something that's occurring now in biology, and you have a ringside seat. How would biology handle an outbreak of something that we've never really given a lot of context to? And this is sort of the best that we can do right now in biology, coming up with these theories as quickly as we can, changing the way that we actually do biology along the way. So I'm going to go and talk to you a little bit about some of the basic biology that is currently in place for the Flaviviridae, and Zika is one of those different types of viruses that are involved in this. And then we'll talk more about specific cases of microcephaly leading up to this paper and this theory that maybe what happens is this virus infects progenitor cells, and these progenitor cells either then die or become smaller, and that accounts for microcephaly. Again, it's a theory. We don't really have a way to prove this well, and it's done in human models, which again is something that I want to get across to you, but we really have no idea if this model is actually correct. So again, just to get a couple of really important facts across to you. Right now, we think that the human vectors are the most important ones that currently exist in Latin America. So if you have mosquitoes and they are biting you and drawing up your blood and then transmitting at the same time the Zika from someone else who's been infected, we think that this is the root of infection. We know that in places like Uganda, originally monkeys were the vectors that were involved, and there's some evidence that maybe rodents are also involved in this process, but we're not 100% sure right now. Once a mosquito is infected, it has a lifetime infection, so mosquitoes usually live between about 10 to 14 days. So during that time, they remain infected, and they can continue to transmit this as long as they are infected. Within humans, we know that the keratinocytes, the cells on your skin, as well as the dendritic cells, which make up the immune system that is part of your skin, at the site where you get bitten, or the site of inoculation, are really important. And we know that through those dendritic cells that take up this virus, this will then move into the lymph nodes and then also into the bloodstream. We'll be talking about this particular factor known as DC sign, which is dendritic cell specific intercellular adhesion molecule 3. And this is part of the CD209 cluster. And this is the way in which Zika virus, when you've been bitten by an infected mosquito, gets into your immune system, and then from your immune system into your lymph nodes, and then from your lymph nodes into the rest of the bloodstream. And this cycle will continue throughout the life cycle of that mosquito that takes it up. And again, this DC sign, as you'll see in a moment, are different types of lectin receptors that are really important in this process. These viruses make use of our own immune system to be able to infect the hosts. And you'll see a number of mechanisms. This is just one out of many mechanisms that these viruses use. So there are a couple of things on here, and I'm not trying to make this more confusing than it has to be. We're actually looking at RNA viruses. Just to point out a couple of key differences here, though. We know that some viruses contain DNA and some viruses have RNA as the nucleic acids that are involved. Some of these are double-stranded, and the RNA viruses that we're looking at, the Flaviviridae type, are the single-stranded RNA type. And I want you to be very clear on that. There are other ones that are really important in terms of biology that I'm highlighting here for you. Many of you will have heard of herpes simplex viruses, and these are the double-stranded DNA viruses. I don't think we're going to have time to come back and talk about them, but I point this out because the cytomegalovirus that I mentioned to you earlier, when you look at microcephaly, in the past, before 2015, when you saw microcephaly, it was either because there was a chromosomal abnormality, which was the predominant major issues that led to microcephaly, or you had a toxoplasmosis infection, which I'll talk about later, or you had a cytomegalovirus infection, or rubella infection, or chickenpox infection, etc. So those are completely different families of viruses. And the cytomegalovirus, which is part of the herpes viridae family, is a double-stranded DNA. So it was completely different than what we've been encountering before. Again, we won't have time to talk about the papillomavirus, but the papillomavirus, again, is one of those double-stranded DNA type of viruses. And the reason why I point this out, and one of the reasons why it might be of interest, at some point in the past, we've encountered viruses off and on in our own genomes, and we have remnants of viral infection in our own genomes. And we know that sometimes some of these different viruses leave remnants that result in things like cancer. And we don't fully understand all the mechanisms that are involved in cancer, but we know that the papillomavirus, if your cervical cells get infected, will definitely lead to a higher risk of developing cervical cancer, and in males too, in terms of penile cancer, if your cells are infected there. So the ones that we're more interested in, particularly for the Zika virus, the one at the very bottom that I'm going to spend more time on. One other thing, just to get this out there, this is a single-stranded RNA virus. The picorna viruses are really important because these are the ones that are associated with the common cold, as well as some more serious disorders like polio, as well as hepatitis. And so they're similar, their structures are similar. I'm going to highlight some of those differences, not so much in today's lecture, but in Monday's lecture, as we spend the last sort of bit on the whole viruses and Zika virus, before we get into molecular biological techniques that might help us to get rid of some of these issues. And the last one that I'll point out is this Toga viridae family, which is that rubella. This is the only other known cause up until 2015 of causing microcephaly. So we know viruses cause microcephaly. There were no reports prior to 2015 that actually having Zika virus, which is a member of the single-stranded RNA positive stranded viruses of Flaviviridae, were actually involved in microcephaly. It was brand new. It was like saying that if you drink Coca-Cola, you are going to end up getting I don't know, smarter or something like that. It was brand new. This association hadn't been made before, even though other viruses were previously associated with microcephaly. Again, I sort of pointed out this to you earlier, that there was a timeline that was in place. It was first reported in 1947, and until about the year 2013, 2014, we really didn't pay much attention to the Zika virus at all. And we know that it gets transmitted from person to person by members of the Aedes mosquito family. So either the Aegypti or the Albopictus. Those are the only two that we know of right now that can actually transmit this. As some of you have read in the newspapers recently, Brock University has actually taken the lead in Canada. They want to know, since one of those family members, the Flaviviridae, in theory, which is West Nile virus, West Nile is found here in Toronto. It's found here in Canada. It causes many of the same symptoms that we have for things like the Zika. Once you are infected with those, can Zika actually survive in a different genus altogether? let me ask you, you don't have this slide, and you can take this information down if you wish. You don't have this slide, but how many different family or genuses of mosquitoes do you think there are? Any guesses? Do you think it's like 2, 3, 4, 5? More than that? Any guesses at all? This is your final exam question. You need to make a guess. What will you guess? Sorry? None of the above. Okay, so at least someone's making a guess. Good. Any other guesses? 2? 3? How many different genuses of mosquitoes are there? Without looking it up. 3? 3. Final answer? Final answer. Okay, great. Actually, it turns out that there are 3. So you'd be, if you're going to sit beside someone and you're not planning to cheat, sit beside eggs here at the front. So which of these 3 do you think is the most common type we have here in Canada and in Toronto? Any ideas? Do we have the Aedes type, in other words? Do you think we have Aedes aegypti or albopictus? We don't. So now you're down to 2 choices. Do you think we have the Anopheles or the Culex type? Any ideas, guesses? Want to try another guess? Anopheles. Well, your streak is over. Nice try. So it's this Culex type is the most common one here. So we want to know, can this Culex type, which has been known to carry West Nile virus, also carry Zika if it moves its way up here? If people are infected and it starts going from one person to another. So someone from an infected area comes up here, gets bitten by a Culex mosquito, which normally carries West Nile. Can that mosquito also get infected and then pass this on to other individuals? We want to be able to know if it can live inside those different mosquitoes. Now, I know some of you have had virology courses, so I won't go into a lot of detail. And the details that you need to know are the more important ones that are on this slide. When we talk about different viruses, which is on the next diagram, and we start looking at viral structures, the most important parts of the virus is actually whether or not it has a DNA strand or an RNA strand. And this nucleic acid, which makes up the most important parts of the virus in this virus particle, is surrounded by these this capsid structure, and this whole thing taken together would be a capsid. Okay, and if it's part of with the nuclear material, with the DNA, it's called a nucleocapsid. So this whole structure that looks like a pineapple is the capsid. The nucleic acid inside plus that structure that looks like a pineapple is called the nucleocapsid, and that tells us a lot about the different types of viruses that we're looking at. And each of these little shoe-like structures is known as a capsomere, one part or one element of the capsid. And why is all of this important? Because many of these different types of viruses, whether they're double-stranded DNA viruses or single-stranded RNA viruses, we can tell a lot about how they infect different types of cells by looking at their overall structures. So again, this is a naked virus, meaning that there is no envelope that surrounds it. And again, we can see that there is a nucleocapsid. So here is the capsid that we just took a look at. Here is the nucleic acid, whether it's DNA or RNA, inside of the capsid structure. And you can see the exact same thing over here on the right in this enveloped virus. And this additional layer is actually really important. Sometimes it is incredibly important because it tells us a lot about the infectivity of the overall virus. It gives us some strategies. It gives us some ideas as to how this virus is being transmitted from individual to individual. So the Flaviviridae, the ones that we're talking about, Zika being one of them, West Nile being another, the Japanese encephalitis being another, all of these envelope viruses have this membrane that surrounds that nucleocapsid. And Zika is one of those enveloped viruses. And these lipid bilayers are really important because they have a number of unique features that allow this to actually be infectious, that allows it to be taken up. It's really this envelope and different embedded proteins that are found on the envelope that help to allow this virus to get inside the cell. This envelope, by the way, is actually acquired from the host cells. So when they start the infection process, they'll actually be using your own membranes as part of their structural elements. So this envelope is really important because it's part of your own membrane system. It has a number of different phospholipids, and these phospholipids and phosphoproteins that I'm going to be talking to you about are really, really important. And I'll just ask you to hold on to this thought for a moment, this phospholipid bilayer being important, as well as the phosphoproteins that are associated with this envelope. There are other proteins that are encoded specifically via the virus genome itself, not from your own genome. And these sometimes lead to different spikes that allow for better adhesion to the host membrane. And I'll get to that in just one moment. And this is really the important part, that different types of envelope proteins and glycoproteins that are found on the envelope, on the outside of these viruses, are how they recognize and infect that initial host cell. And we just talked about at the beginning that DC sign on those dendritic cells within your skin are the first sort of players in this infectivity. So there are lots of different mechanisms for how viruses enter into the cell. The simplest type is where we have this phage genome, and we won't talk a lot about this. This isn't a mechanism that Flaviviridae uses, where it directly injects the viral genome into the cytoplasm of the cell. And we're not going to be talking about this. The other two types actually are very similar to what happens in Flaviviridae. And in particular for Flaviviridae, we're looking at the type that actually involves endocytosis. The recognized machinery, the protein complexes that actually remove something from the surface into the cytoplasm. And this is the mechanism up here on the right corner labeled as C, the endocytotic mechanism that Flaviviridae is using. And you guys all have heard about endocytosis in your first year biology classes, right? So you know all about the different machinery that's involved in this process here. So there are some specific structures that I wanted to mention to you in this particular Zika virus, and we have just recently learned a lot about the structure of the Zika. For the longest time, because it wasn't a very common type of viral disorder or a virus that infected too many individuals, people just didn't care. It was something that was going on in Micronesia. It was something going on in some small islands. People really did not care a lot about the overall structure of Zika, and it's only in the last little while that the structure of Zika, outside of what we knew about dengue fever, which is another Flaviviridae family, that we started to know a little bit more about it. In fact, it looks a lot like dengue. And so we know that there are specific proteins that are found on the outside of the cell, and these E proteins form a dimer, as you see here, and they're the ones that are coating the outside of the cell that you see here. And they form these dimer-like structures that will be rolling and trying to make contact with different cells as they are getting into your bloodstream. There are other proteins that are found underneath those, and these M proteins are really important, and I'll talk about those in a little bit. And then the C proteins are found in the cytoplasmic region, and they're thought to interact with the genomic RNA in this particular scenario. These are really small. Overall, they don't have a icosahedral structure. They are spherical under an EM microgram. They're very small, about 50 nanometers in diameter, and overall, it's a round structure, but the individual elements do have some structure that looks like a polygon when you take a look at the overall way in which this soccer ball comes together. So if we're to take a look, why are these different proteins, the E proteins, why are they so important? There are factors that are involved in this. So as this virus is encountering different cell types, so it's been ejected as the mosquito is biting you, the mosquito has released all these viral particles under your skin, and in your skin, it's encountering all sorts of different cells. Some of those cells are immune cells. Some of those cells will be keratinocytes. Some of those cells might be cells that are not associated with the immune system, etc. And one of the things that they try to do is to attach on, to make use of the endocytic machinery. They need to be able to attach to certain receptors that are found on the host cell. So again, this is the host cell down here. Here's the viral particle. Those E protein complexes, those dimers, are trying to grab onto something. They're trying to latch onto something to help them get into the cell. There are all sorts of different molecules that different viruses use. There are heparin sulfates, there are other membrane proteins, there are sialic acid modifications on different proteins that are found on the host cell. And if it encounters the appropriate receptor on the appropriate host cell, then it starts to latch on. And once it's latched on, it will use that endocytic machinery to enter into the cell. That's one of the mechanisms, okay? And we'll talk about that again in a moment. There is another mechanism at play here as well. So when this entry receptor that we're going to talk about in a moment, and I've already talked to you about this being that DC sign being one of those, that will then cause endocytosis to occur, there is another sort of mechanism that comes into play that makes use of what is written here on the right. That in addition to all of these different glycoproteins and glycolipids, the virus itself starts to engage in a little bit of molecular decoy. And I think it's actually on the next slide. So I'll get to that in 1 second. So these are some of the other sort of viruses that make use of exactly the same mechanism. I've talked to you about DC sign as one of the receptors that are found on our dendritic cells that allow for entry. Related members of Flaviviridae, like West Nile and dengue, use that same E protein envelope protein, the same sort of dimer system, but they bind to very different surface molecules, okay? And we know for the Zika, it's that DC sign that seems to be really important. And you can see that many of these are sort of sticking out of the cell as integrin proteins, etc., and that once they bind to the appropriate entry receptors, that this causes them to move into the cell through endocytosis. And again, those are important mechanisms. You need to have a receptor, an entry receptor on your host cell. For Zika, it happens to be the DC sign, and then it's all through clathrin-mediated endocytosis. There's one other mechanism I told you that is really important. It goes back to the earlier slides that I mentioned to you at the very outset. So once that viral envelope protein E that's found on the virus binds to DC sign and it initiates this process that causes endocytosis, there's one other thing that it starts to do. It also engages in what is known as apoptotic mimicry. Have any of you heard of apoptotic mimicry before? Do you know what that is? Do you know what apoptosis is? Okay, so apoptosis is a form of cell death. And in that form of cell death, we don't really have a lot of cell remnants because the cell that's dying gets phagocytosed by nearby neighbors. It gets engulfed by other cell types. And that's one of the reasons why we don't see a lot of remnants of cell death. In order for that engulfment to occur, there has to be something special on the dying cell that allows it to get engulfed. Do you know what happens? Have you heard about it in your biochemistry class, for example, or in your second year bio class for apoptosis? So one of those phospholipids, do you recall what happens to it? I'm not sure what this means, but I think you're right. But you can't put that on a final exam. It'd be really cool, but wouldn't work. Anyone want to translate this? So one of those phospholipids, phosphatidylserine, the phosphoserine actually gets turned inside out. It goes from being exposed on the inside to now being expressed on the outside. When we start to see phosphatidylserine on the surface of a cell, that is a sign that that cell is dying, and it's a marker for that cell that is dying by apoptosis. This virus decides that it wants to look like it's dying. It wants to help this machinery to be endocytosed into the cell. So it has a receptor That's one form that it uses, and then it engages in this apoptotic mimicry that allows for phosphatidylserine to be expressed on the cell surface that helps even more to get that cell internalized through endocytosis. Eventually, once it's inside the cell, inside the cytoplasm, it will start to fuse with the endosomal membranes, and that viral genome starts to get released into the cytoplasm. And that positive sense, and I mentioned that this is the characteristic feature of Zika and the Flaviviridae family, that positive sense genomic DNA, ssRNA, single-stranded RNA, then gets translated into a polyprotein, a big protein that will then subsequently be cut into individual pieces like the E, M, and C proteins that we saw earlier in order to produce the overall structure of the virus. We need to be able to propagate the entire genome of the virus, and so replication also occurs at the surface of the endoplasmic reticulum, and that double-stranded genome is then synthesized into the genomic SS single-stranded RNA form. And this allows then for this whole process to be replicated. It's already produced the larger polyprotein, which has been cleaved up and includes some of those structural elements that form the capsid. It will then combine together in the cytoplasm, and then it will be packaged and ready to be released as it gets transported up into the Golgi apparatus, and then it gets released through exocytosis. And this is the whole sort of series that I just showed you on that previous 2 slides that gets into how it enters into the cell. And you should make sure that you understand the factors that are found on the virus itself. So there are 2 that we talked about, the envelope proteins, the E proteins, and how they interact specifically with the host receptors. And in this case for Zika, it happens to be the DC sign that's found on the dendritic cells. And then following that, once it binds, in order to help it get into the cell, it engages in apoptotic mimicry. It flips around the phosphatidylserines. It gets expressed on the outside. And once it gets internalized through endocytosis, the steps that I just mentioned to you allow it to then get repackaged in the cytoplasm, and then it gets released from that infected cell into other cells, which will then start to propagate it. Many cells will start to become infected, and that just really exponentially increases throughout other cells within the bloodstream. Okay, so again, as a mosquito bites, it will release into your skin. It will start to release. In this case, it says dengue, but you can imagine this is Zika. It will infect through specific receptors these dendritic cells, and this dendritic cell here called DC, this dendritic cell has that DC sign molecule associated with it. As it gets infected, it will then start to transmit these to other activated macrophages, and together, as they move through the lymph nodes, they'll start to infect other cells that will enter into the bloodstream as well. And this propagates throughout your bloodstream, and the next mosquito that comes along will encounter the same sorts of infectious viral particles that release from these infected cells, and this propagates this transmission cycle that's shown to you here. And don't worry about all of trying to read all of the details. It's just to give you an idea of how this transmission works, and this allows for this to spread from one individual to another every time a mosquito happens to bite. Do you have any questions about this before we go on? Okay, let me switch gears just a little bit. One of the things that makes this really, really scary and really, really interesting at the same time is this simple statistic down here. In 2014, the year before this outbreak occurred in Brazil, when you track in using epidemiology, and it's reported by the different hospitals, when you track the number of cases that are due to chromosomal abnormalities, other infections, etc., when you track the number, it was 147. Now, when you track 1 year later, the number of cases of microcephaly that were being reported, the number ballooned into about 3,900, almost 4,000. That is a huge increase. That is not possible by statistics alone. Every single year before that, it was generally somewhere between 100 to about 200 cases. And now we're running into the thousands. This caused a lot of concern where no one understood what was going on. And there's still a lot of controversy as to what actually is causing this outbreak of microcephaly. So microcephaly is usually something that can be diagnosed as early as the first trimester. And in fact, if you think that there is a possibility that the child that you're carrying has microcephaly, you can get tested in the first trimester. And there are screening methods, and then there are diagnostic methods. The screening methods are ones where you're simply measuring through a number of different tests, things like an ultrasound, just seeing if the size of the head looks like it should be normal for the first trimester or so. And it's also looking for other things. So the ultrasound is typically looking not only at the size of the head, but also for extra fluid at the base of the baby's neck, because this increased fluid is one of the early signs that actually leads to microcephaly, and is typically associated with a chromosomal disorder, or it might be associated also with a heart defect. And that's a screen. It's done initially, and then it gets into a firm diagnosis. You can also be measuring different types of proteins that are found in the maternal bloodstream, like human chorionic gonadotropin, as well as pregnancy-associated plasma protein. And if these are abnormally high or abnormally low, and they're outside of the normal test ranges, it might mean that for either chromosomal aberrations or for other reasons, it could mean that there is something going on like microcephaly in this child. And these are typical screens that are done. For example, in older women, in women that have recently had an infection of chickenpox, if you suspect that there is a genetic background in these individuals, these types of screenings will be done in the first trimester between weeks 11 and 13 during pregnancy. If that test happens to come back as being abnormal, then there are other diagnostic tests that can be performed that I'll get to in a few moments that will further help to define whether it's a heart defect, if it's microcephaly, if there's another type of disorder that's going on. And again, there are reasons why these diagnostic tests are done. So if you're a slightly older individual over the age of 35, it's typically done because of potential trisomy effects. If you've had a previous pregnancy with a birth defect, you're often given this diagnostic test as well. Chronic diseases, if you have diabetes, if you have lupus or epilepsy, if you're on certain types of medications, you go through this screening process. And again, when those first individuals who are pregnant and were infected with Zika, they had no reason to suspect. They weren't 35 years of age. They had no medication, no other issues, no chromosomal abnormalities, all young individuals. There was no reason for them to get screened. There was no reason to suspect anything was going wrong in those individuals until they gave birth. So other types of diagnostic tests that can be done is this chronic chorionic villus sampling, or CVS. And the villi is part of the placenta. It is not part of the developing embryo. So there is no harm at all to the embryo or to the fetus. It is done as part of the placenta, and a small portion of the placenta is then removed, and it can be checked for either chromosomal or genetic aberrations. And again, I'll go through that in some more detail in about 2 slides. And again, it's usually done. It's not something that's typically done. It's only done if there's an abnormality on those first trimester screens. So your APAA levels are really high. There's something wrong with some of the other tests. Your ultrasound looks like there might be some issues with it. Then you're usually referred for this chorionic villus sampling. And again, it's usually done at around the same time, between weeks 10 to 12, so very similar to the weeks 11 to 13 in that first trimester. So other forms of testing include amniocentesis. So the amniotic fluid, you can withdraw some of the amniotic fluid, typically done for things like Down syndrome, cystic fibrosis, Tay-Sachs disease, etc. You're measuring for specific proteins like AFP. And higher levels of AFP might mean that you have an opening in your neural tube, which might mean that you might have microcephaly, or you might have other neural related deficits, and you measure these from the amniotic fluid directly. Can also measure for acetylcholinesterase, and that acetylcholinesterase is normally kept inside the nervous system and is only found in the nervous system. And if you find it in the amniotic fluid, it means that there's a way for it to get out, and it usually means that it is an opening in the neural tube. So as it forms the brain, as it forms the spinal cord, the neural tube is usually closed, and if it happens to stay open, it's usually not a good sign for the developing embryo. So again, this is sort of the amniocentesis. The only issue with amniocentesis is that it's done a little bit later. It's usually done between weeks 14 to 16. When there is less chance that the amniotic fluid withdrawal is going to harm the developing fetus. And again, the cells and the fluid themselves can be examined for genetic abnormalities. So when you withdraw amniotic fluid, you not only get the fluid that allows you to check for acetylcholinesterase and AFP, you can actually get fetal cells that are released. You can take some of those cells, grow them up in cell culture, and then engage in karyotyping, which allows you to see if there are any chromosomal abnormalities in the developing fetus. So all of these things are different diagnostic tests that can be used to help us in determining if there are going to be issues with the developing fetus. None of those were frontline tests that were done in Brazil until very, very late. It wasn't done until fairly recently, as a matter of fact, for screening for microcephaly, because we had no idea what was going on. One of the reasons why we think that Zika virus is actually responsible for this is that in this amniotic fluid, there have been some suggestions that Zika viral particles, the types of particles, the virions that I just showed you, and some of the proteins that are associated with Zika were found in the amniotic fluid. Now, if the mother had never been exposed to the Zika, etc., there shouldn't be any in the amniotic fluid. And in addition, in 2 cases in Brazil, within the brains of babies that were born with microcephaly that didn't survive, in the brain tissue itself, there were remnants of the viral particles found in the brain tissue of those babies that had microcephaly. So microcephaly itself normally has a number of issues that arise from chromosomal abnormalities. You might have trisomy, not just Down syndrome, but trisomy of the 13th chromosome or the 18th chromosome. You might have deletions of the short arm of chromosome 3 or chromosome 5, etc. And there are a number of other sorts of chromosomal abnormalities that have been linked with microcephaly. None of those, by the way, occurred in Brazil. None of the individuals that were tested for this had any of these chromosomal abnormalities. There were no problems that were identified related to decreased oxygen or cerebral anoxia. And again, as far as anyone could tell, they had none of these other infections either. The only infections they had were Zika. They didn't have infections with West Nile, dengue, Japanese encephalitis, none of those traditional cytomegalovirus, etc. Types of infections. It turned out that Zika was the only thing that they were exposed to. So microcephaly, it can either be an isolated condition. There are no other complications. It could be you have microcephaly because you have Down syndrome, and in addition to that, you might have congenital heart defects. You might have some other issues as well, but microcephaly itself can be an isolated condition. That's the only thing that's wrong with the baby in terms of a birth defect. Usually microcephaly is defined as the head circumference being smaller than 2 standard deviations than a normal sex matched head circumference of a normal baby. Okay, so 2 standard deviations below. In other words, really, really small. In some instances, when it's 3 standard deviations lower, it's called extreme microcephaly. There's almost no cranial vault whatsoever. Do you know when this measurement is usually done? Is it done immediately at the time of birth? No? When do you think it's done? Any ideas? 1 week of age, 2 weeks of age, a month? Can just imagine you guys on the final exam. You come across a question and you're not 100% sure. You're going to make a guess, right? Why not make a guess now? Sorry? 1 week? A year? Hasn't been a year in Brazil, not quite yet. Almost, we're coming up to 1 year. So it's usually done about 24 hours later. And again, I'm hoping some of you will actually take me up on my offer to get out into the hospitals, see what goes on in the OR, see what goes on in the NICU, etc. And I hope that you actually get to experience some of the things that we talk about. I remember when my kid was being born, and I was in the delivery room with my wife, and I was seeing that my baby's head was coming out, and I was going, wow, this is really, really cool. And my wife, but the other thing that went through my head at the time was, wow, my baby's going to be about like this tiny, because like the only part of the head that you can see is the back part, the crown of the head. And it's like one thing to actually see a baby being born. It's another to actually hear about a baby being born. Not that I want you to rush out and have babies or anything else, but it's a really different experience. And I think the one thing that I realized from this, that you measure the size of the baby's head a day after, because when the baby comes out and it gets compressed and then it gets pulled out, that baby has a really misshapen head. And it takes time for it to kind of go back to where it's normally shaped. And usually the head size and the circumference is measured about a day to a day and a half later, once the baby sort of recovered from the trauma of delivery. So we know that if you have microcephaly, it is a lifelong condition. That baby is going to have seizures. There are potentials for seizures. I mean, the brain isn't fully developed, and as a result, it might have all sorts of seizures. There are going to be all sorts of developmental delays in terms of walking, being able to talk, etc. Obviously, intellectual disability, being able to function normally for the rest of their lives. There are going to be issues associated with movement. Everything here that you see is related to the brain. Feeding, hearing loss, vision, all related to the brain, because the brain simply doesn't develop the way that it should. So this is actually taken from the CDC website. Most of today's lecture is actually taken from the CDC website, which updates every 24 hours. And this is what they say that we know. Right now, that pregnant women can actually get, who are infected with the Zika virus, likely through infections by mosquitoes. It has actually been shown that the Zika virus can actually survive in semen, and it survives in semen much longer than we thought was possible before. And so it can be transmitted sexually. And again, a pregnant woman can actually pass the Zika virus along to the fetus. The one thing that we are also aware of that the CDC made a strong note of, a few weeks ago, there were a lot of news reports coming out of Brazil that said that maybe it was actually the chemical compound that was used to stop mosquitoes from being able to reproduce that was actually causing Zika. And this chemical, pyriproxyfen, is actually not the cause of Zika, and the CDC has come out very, very strongly saying that this chemical is not the cause of Zika. They can say that with absolute confidence because this chemical has been used in Brazil and in other parts of Latin America for over 10 years. And that the use hasn't increased, nor has it decreased, and it's stayed the same. And overall, they know that this chemical is probably not involved. So we're now interested in, how does this actually work? How does this Zika virus, if it infects, and we're not talking about dendritic cells, we're talking about cells in your brain, how does it actually cause microcephaly? Well, this is actually really interesting too. I don't know if you are aware of how science works, but one of the things that we try to do in science is really, really competitive. We talk about people trying to get out these big papers in these big journals all the time, but Zika has actually fundamentally changed the way we report science. Before, what happened was you didn't want to let other people know what you were working on. You just wanted to spring this brand new paper on them that they would be reading online as part of a published journal. Now, because of Zika and because of the way that it's spreading, more and more of the reports are actually wide open access. They're publishing their results from their labs before it actually gets peer reviewed. And this is one of the first papers that actually did that. They put all of their lab results way back in January all online. They wanted other scientists to see what they were doing, see what their results are. And basically what they said was Zika doesn't only infect the dendritic cells. It can actually infect neuronal stem cells, or NSCs. It can cause those neuronal stem cells to undergo apoptotic cell death. It can cause the colonies that normally form in the brain in culture called neurospheres. It can make those neurospheres smaller. And when these neurospheres are organized as mini brains called organoids, brain organoids, the brain organoids themselves are smaller. So this is one potential mechanism by which the Zika actually causes this to occur. This other paper that was more formally published that came out not very long afterwards was actually a really important paper because it confirmed that initial sort of publication. And Guoling Ming has actually shown very, very similar results that we're going to go through in some more detail next week, that actually suggests that if you take skin fibroblasts and you use the Yamanaka factors to make them back into stem cells or pluripotential stem cells, and you then convert them into neurons and you infect those stem cells with Zika virus, that they do exactly what that other paper said, that they grow smaller, or they start dying by apoptosis, or there's gene dysregulation, and the number of neurons and how the neurons form are very, very different looking. So since we know that Zika can infect neurons, it's found in the amniotic fluid, and it does potentially have a link with microcephaly, this might be the beginnings of a mechanism that has really interested scientists, because this is a brand new sort of field for them. This is the first time that they're actively trying to find a mechanism for microcephaly and trying to race to find out how to maybe prevent microcephaly from occurring in places like Brazil. And we'll talk about this in more detail on Monday. And starting next week, we don't have a class on Friday. So starting on Monday, my goal is to try to get you ready for the final exam. So at the end of the last section on Monday, I'm going to take you through some practice problems, and we'll leave them up there. And I'm hoping that you're going to spend time in the next 2 weeks getting ready for the final exam. Because if I'm not mistaken, your final exam is really, really early, right? It's like April the 13th or something, and our last class is sort of that week before then. So you need to start getting ready for your final exam early. Please let me help you if I can. And for those of you presenting next week, good luck.